So hello, welcome back. You hear me well? Okay. So it's my pleasure to be here finally. And it's my pleasure to chair the summary breakout session. We have three interesting topics dealing with the heart, kidney, and liver. And we start with the heart.
Right, thanks. Okay, great. The timer's on. Well, wonderful. Thanks again for everyone who's made it back for the second day. My name is Raj Gupta. For those of you I haven't met, I'm a cardiologist at Brigham and Women's Hospital and a researcher until recently at the Broad Institute, but now at the Brigham. And I've sort of come to single-cell analysis differently than maybe a lot of people. Sort of interested in genomics and sort of was using GTEx data to identify EQTLs that were relevant to disease. And in that, we saw that cellular heterogeneity was going to be important to explain. And so, that's my background. And we had a great session for the heart and vascular session here in the auditorium. We had a lot of people who came. And I'll say that right off the bat, there was a little controversy over the statement, but I do feel that the heart and vascular are two entirely separate organs, right? So it was a bit challenging for us to sort of talk about both, given that they both hold blood or come in contact with blood, but beyond that they have different cell types, different diseases, and so we sort of kind of wandered through talking about both of them. But I'll say that at the beginning, to summarize our session, we had the benefit of a really great audience of people from around the world, all of whom are interested in single-cell analysis of the heart or vasculature in some way. I'd say it was about 90% heart, admittedly. I'm more of a vascular person, so feeling a bit lonely. But complementary efforts from a CZI consortium, the University of Chicago, Europe, Asia, Australia, and then even here locally there were some groups. So great representation, and there was a lot of interaction on some of our challenges. And so before we sort of went into the complaining about how this was not going to work, we sort of summarized the state of the field for the heart. And we say there was two major themes, I think, at this point, though this is rapidly changing by the minute even. But first, there have been a lot of papers that have found disease-relevant subpopulations in animal models. And the animal models have been incredibly informative because there's less intra-individual variability. The disease processes are more exact, I would say, than we see in the human. And so there have been some interesting studies there. And then there have been some recent insights from developmental biology. And this is in human in many cases. So just to summarize, this is probably a review for many people here, but a few papers that showed disease-relevant subpopulations. This was a paper that was published this year in Circulation at the top, where they took ischemic mice. So they induced ischemia in mice, and they got ischemic regions of the heart and did single-cell sampling. Sorry, here. And what they found is that there was this unique subpopulation of cells that had mostly red dots, which were from the ischemic mice, the 3 ischemic mice. And they found that that population expressed more CKAP4, which they sort of identified as a new modulator of fibroblast activation in ischemia. So, the next step would be to validate this in human studies, right? Is that same population? Are those same genetic modifiers relevant to human ischemic disease? And then sort of a second great paper on the vasculature, also in mice, was from a molecular atlas of cell types and zonation in the brain vasculature. And that was in Nature, where they were actually showing that at different stages along the vascular tree, all the way from big vessels to medium-sized arterioles to capillaries, then back to the veins, there was a gradation of gene expression, suggesting that the vasculature could sort of serve as a signpost for different parts of the body. So that's the sort of disease-relevant subpopulations from animals. And then there were some recent insights into developmental biology that we had a representation of these groups in the audience. It's from Stanford. They recently did a single-cell analysis of progenitor cells in coronary artery populations. And then right off and not even hot off the press, not even made its way to press, is a paper from a group in Sweden where they did expression analysis in developing human hearts. And by developing, they say sort of embryologic age 6 to 12 weeks, which I find fascinating, and we'll sort of delve into that paper more. So the challenges that we discussed in this session were many and a bit overwhelming at times, but I'd say that they were probably similar to what the other groups discussed as well. Maybe a few things are unique to the heart in particular. So first of all, we spent a lot of time talking about standardization of tissue procurement, sampling, and dissociation protocols. And so that doesn't really exist for the heart yet. I think, from our audience, just of the people here, there were maybe, at least 10 groups who are working on this. And we haven't discussed, are we going to use nuclear sequencing or cellular sequencing? Are we going to use certain regions of the heart? So I think that right now there's no standardization, and that's sort of a big task, I think, for our working group going forward. And again, sort of similar to other organs, that'll be the case. But for the heart in particular, there's no sort of repetitive structure, right? So, at the last point here, as I say this, it's a non-repetitive organ. So there's not like zones of the heart. Every cell in the heart could be different from every other cell. The apex could be different from the septum, even within the same anatomical structure. And for that reason, we might have to be more exact about our standardization. We might have to have better coordinates so we can directly compare cells. Or we could sort of say, well, forget it. It's going to be too hard. We'll see what comes out of it. And I think our consensus was that we should be more exact. I think that was the consensus from our group. And I'll talk about that in a second. And then, the second sort of thing is this problem of inter-individual variability. So what metadata would we collect? So certainly disease. So, and we'll talk about this. There's actually no such thing as a normal heart that we will obtain. So even in these sort of cases of brainstem death donors who have presumably normal hearts that are then rejected for organ transplant, even those hearts, because of brain death itself induces some microvascular ischemia, some coronary vasospasm. There will be microinfarcts in those tissues. There will be diseased segments. There's always subclinical atherosclerosis in anyone over the age of 20. So we will see disease in these tissues. So we'll have to think of variables to record so that we can then sort of after the fact determine that. And then obviously gender, ethnicity, and sex. And there's other sort of clinical markers we could record as well. Inflammatory markers, lipid levels. But we thought these would maybe be the 4 things, at least on a first pass, to focus on. And then I sort of mentioned this tissue access. So we spent a lot of time on this as well. And so if we were going to have a standardized way of doing this, if there's no such thing as a normal heart, how would we sort of account for this? And we thought, well, you could either go with post-mortem samples or clinical biopsies. Clinical biopsies are pretty rare from the heart. We would never biopsy a normal heart. And even the diseased hearts or post-transplant hearts that we biopsy, we take such a small piece of tissue and are never really sure where that biopsy is from. The way it's done is that you sort of snake a catheter into the internal jugular vein into the right side of the heart and just sort of blindly use these forceps to grab little biopsies of tissue. It doesn't sound so inspiring, I guess, to describe it like that. But I think that would be a very difficult tissue source for this kind of community to standardize. So we focused on these post-mortem hearts. And even though I sort of told you all the problems with using rejected organ donor hearts, that did seem to be the best sort of first pass for our group. And so we just have to account for preclinical disease. And so we can think about ways to do that. And that might be collecting medical record information, and it could be pathology samples. So at the same time that the heart is taken for single cell, it'd be important to also use some sample for formalin-fixed tissue and look at pathology so that if we see sort of variability, can we go back to the slides and see if there was more fibrosis or more hypertrophy and those markers of disease that might not have been clinically apparent but will be pathologically apparent. And so, and then for vascular tissue, again, I'm a little jealous. The heart got 90% of the discussion, and I'm sort of more interested in vascular tissue. But I would say this was easier. Maybe that's why we didn't talk about it. But for vascular tissue procurement will be easier in some ways because we do biopsies from different parts of the vascular tree. Aortic biopsies occur during surgery. When we take out the aortic valve during aortic valve replacement, we get some aortic tissue. And then also there's this sort of opportunity/challenge of there will be sort of residual vascular tissue that comes along with everyone else's organ. And should we have like a protocol for studying that? I mean, everyone will use different protocols for tissue isolation. But I think, as a vascular interest community, I think that could be one of our main challenges over the next year is to think of a way to take all the endothelial cells that are obtained from other tissues and reanalyze them in some sort of meaningful way. And so then, So specific challenges to the heart. And so this actually I found to be quite interesting is that, fine, let's say we can dissociate the tissue and we can sort of have a standardized specimen and a standardized protocol and a sampling region that we're comfortable with. The problem is that 10% of adult cardiac myocytes have at least 2 nuclei. So even if we're using a sort of multi-nuclear, or we're using nuke-seq as our standard protocol, we would probably reject that 10% of cells. And these cells might be very relevant to regeneration or disease progression. And so that's sort of a challenge that we might have to deal with, is that how do we sort of capture multinucleated cells if we're using nuke-seq as our standard protocol. And then again, cardiac myocytes are only 25% of heart cells. And so some of the challenges that people voiced are that cardiac myocytes, using cellular analysis, is almost impossible. They're too big to sort of use through a standard droplet 10x system. But perhaps we could do nuke-seq with some cardiac myocytes, or but a different protocol for other cardiac myocytes. I think for me, this sort of emphasized that if we're going to have a comprehensive atlas of the heart, it's going to have to include many different dissociation protocols. It won't be sort of one protocol that works for everyone, is sort of my sense from some of these challenges. But it wasn't all sort of doom and gloom. We saw a couple unique opportunities as well. So first of all, we spent a little bit of time talking about if vascular cells could serve as a common coordinate framework for the whole body, right? There's recent papers from the Tabula Muris group where they showed that the transcription factor signature for endothelial cells was representative of the organ that they came from. So that sort of suggests that there is a signature. The problem is endothelial cells are also pretty plastic, so even 2 cells next to each other, if they're exposed to different flow conditions or different lipids, they might be different. So we don't know if this would take some method development and standardization, but if any cell could serve as a common coordinate framework, it would be an endothelial cell or a vascular muscle cell or an adventitial fibroblast, and so it's worth exploring. I'd say that the heart and vasculature, the diseases of these organs are of cell type and cell state. They're patterning defects. Congenital heart disease is certainly a developmental defect, and the efforts to look at embryos is going to be really informative there. Vascular diseases like atherosclerosis were sort of the first cellular diseases, so we're sort of excited about that. Other organs also are impacted by their vascular effects, and there's kidney, lung, and gut and brain vascular diseases. Sort of a challenge and an opportunity is that vascular cells contact blood and so integrate the environment into their transcriptome. So how do we account for that? If we're going to get vascular cells from 50 donors, should we know a little bit more about their exposures or their lipid levels or those kinds of things, because that will be reflected in their single cell profiles. And then, like I said, vascular cells are abundant, and cell-cell interactions are really integral to this biology, like the endothelial cell immune cell interaction. So to kind of end, we sort of tried to postulate a little bit what a first draft atlas would look like. And so we sort of settled on the first draft atlas for the heart would definitely be limited, we think, to post-mortem, brainstem death, unsuitable for organ donation hearts. And that, maybe that's just our opinion, but we thought that that was the best case of sort of a standard tissue source that would be as close to normal as possible. And then, we sort of, in the HCA white paper, I think it was postulated that they would use 20 normal hearts. And that could account for some inter-individual variability, but the truth is we have no idea. So I think it would be important to have an iterative experimental design, important to sort of freeze samples for pathology or for subsequent analysis and maybe reanalyze those samples at later times. But in the first 20 hearts, I think the main question would be, what is the inter-individual variability? And is our metadata capturing that? Are our sort of recordings of age, sex, gender and disease state, is that adequate? And then sort of I liked what the lung group said yesterday, is that the disease state actually could inform the normal instead of the other way around in some cases, because every heart's going to have some component of disease, and we don't know what that is beforehand. So in looking at sort of the extreme versions, like extreme cardiomyopathy or extreme atherosclerosis, we could use those signals to inform what is the extent of cardiomyopathy or atherosclerosis in the so-called normal heart. And then finally, extensive histology to validate markers and spatial location, I think, will be important. And then finally, the vasculature, I think a first draft atlas would be something like a large vessel, and then medium and small vessels would be sort of analyzed separately. And a large vessel would be surgical biopsy specimens from an aorta. So these wouldn't be postmortem samples that could be obtained. It would be relatively easy to get something like 50, 100 samples from the aorta. But medium and small size vessels will have different signatures, and those could be reanalysis of sort of tissue that we get from other single cell efforts, like the kidney, the lung. And all these cells and all these tissues will have some degree of atherosclerosis and variability from exposures, and so we'll have to account for that. And so the final thing I wanted to say is that spatial orientation was obviously a main topic at this meeting in general and in our small discussion group. And so we thought about different ways to do this. And so one way would be, you take the heart and you record distances from major structures, right? You just become very exact about this, 1 centimeter from the aortic valve or something. And that might be good. We pointed to some problems with that. Human hearts are just going to be very different sizes. So we could, one of the pathologists in our group suggested that we take a transverse section across the whole heart. Everyone just finds the midpoint of their heart sample and just goes all the way across and captures everything at that midpoint site. And that sort of was appealing because it could be something that could be standardized. We thought common coordinate frameworks would be hard to develop in human samples, but we could develop them in animal models, and the pig is evidently the most sort of reproducible animal relative to the human heart, even more so than, I guess, primates or rodents, certainly. And so that could be one possibility. And then we could also finally use preclinical imaging, like echocardiography, to assess for normal and diseased regions, and then sort of overlay that with the data that we get. So those are the points we wanted to make from our group, and we're happy to answer any questions.
So great summary, and glad you had a good discussion. I agree that the heart and the vasculature are two different beasts that we evilly and maliciously put together on you. But I actually, I'm a big believer in the vasculature as a possible common coordinate frame. And I think that computationally, again, one would have to be smart about it, not look at the genome-wide distribution, but find the markers that can help with this. And, I do think there's a pretty mature idea that the immune cells need a separate protocol, and the immune atlas is going to collect from everyone and help everyone collect better immune cells from their own organ. But I actually think that the vasculature should present similar protocols, and we can get endothelial cells. And there's also, for example, the strong signature of just how big that organ is to where you are, how big the vessel is, as well as how far each individual cell is from the vessel, creating metabolic gradients that could explain a lot of the gene expression once we get into the spatial data. So I think both as where you are globally in terms of how wide your endothelial cell is, which is something I do think we can pick out of gene expression, and once we get spatial, how far away are you at little microcellular resolution from these vessels, determining gradients, is something extraordinarily powerful that the entire community should think of. So I actually want to punt my question to the other organs. How excited are you about using this as another piece of a common coordinate framework to interpret the data? And this is actually framed at the other organs.
Hi, I think this is a great idea, and we should definitely try and do it. The question is, how do we do it along the way as we acquire data?
So again, my idea would be that there would be some common group of both technologists to figure out how best to actually get these cells, because they're actually pretty tricky, finicky cells, to share protocols with the atlases, as well as computational people, who I know one sitting next to me is very interested in doing that, about developing computational methods to help do this. And we think it could be of great benefit if right here and right now people really think of this as another way to get some coordinate framework inside each and every atlas. But it would require sort of a centralized group, both on the technological and the computational side, to really facilitate each organ, if the organs are excited to play along.
Yeah.
Some more comments, questions? If not, we move on to the kidney. I think you forgot to say that heart is the most important organ. So next are Anna and Sam reporting on the kidney outbreak.
Okay. Hi, everyone. My name is Anna Greka. I am a kidney doctor at the Brigham and a scientist at the Brigham and here at the Broad. And I'm here with my partner in crime, Sam, to talk about kidney. So by way of a friendly competition, yesterday's folks were trying to sort of think about who has the more important organ. Well, we have the cooler logo, I think, and that's what I wanted to compete on, but this is actually the work of Catherine Blakesley, my group, so she's the artist, not me, and we just thought it would be cute to put it in the shape of a kidney. So this is what we talked about today. We had a great discussion. There was a lot of active participation, and we basically broke down the discussion and the project essentially in multiple pieces, and so Sam and I are going to alternate reporting out what was discussed. I just want to invite anybody who was in our session to jump in and keep us through if we are missing something major from the discussion. So just starting from the very beginning, we decided to just go around the room and catalog what activities may already be happening in the kidney atlasing space. And so there are actually some projects that are going on, and in some ways there was a lot of foresight on the part of the NIDDK to start to fund some projects looking at the single cell characterization of diseased kidneys. And so there's an effort called the Kidney Precision Medicine Project, which was represented by Matthias Kretzler here today. There's also an AMP program, also partially funded by the NIH, that also looks at some diseased kidneys. So KPMP is more focused on acute kidney injury and progressive kidney disease, which the most common form of that would be diabetic kidney disease, but just more broadly, chronic kidney disease. And then the AMP is focused on lupus nephritis primarily. But there are other efforts going on around the country. So there's some efforts going on at WashU, and Ben Humphreys was here today to tell us about that. UCSF, Mini Sarwal was here. We have efforts going on at Jackson. Of course, we have ongoing efforts here at Broad as well. All of us trying to work toward a common goal of understanding the human kidney atlas.
So we spent a bit of time discussing what experimental strategies we may take, and discussed also what hasn't worked and what is more promising. There was clearly, everyone felt there was a need that we need some sort of standardization in terms of SOPs. We need to find a reference, an anatomical reference framework of how we take our samples and annotate that, and then also think about feeding that into the HCA database so that it becomes feasible to compare data sets. We had a discussion about frozen versus fresh, and there was a general feeling that sort of, I mean, they've obviously both got their biases, but they both work, and fresh is obviously, it's not necessarily the gold standard actually, but perhaps it's a starting point, and then frozen also will enable us to interrogate very large cohorts and also start thinking about disease. Yes, and then another thought that came up right at the end, and which we might get to, is to perhaps have a benchmarking exercise where a kidney is taken, biopsied let's say in the cortex several times, and then sent to different centers. People do their own sequencing, their own nuclear RNA sequencing, and then annotated, and then we can compare notes at the end to get a sense of how robustly and consistently we can generate and annotate data.
Yeah, and so then we kind of address the next part of this, which is, of course, the computational piece. So assuming that we resolve the experimental part, how do we integrate all of these data across different sites as well, and then also integration of metadata, which I know many of the groups have been struggling with as well. As with every other group, we had the discussion about cell types and cell states and the idea of sort of consensus markers in the community. And I think at the end, the only solution to that is to just continue to do this in an iterative fashion and draw from the experience of many different groups as we try to kind of arrive at our understanding of this. In many cases, a lot of our markers are coming from other from other species or from other modalities, but we can learn collectively and apply them as we try to truly map what a normal human kidney is comprised of. There was a point made, I think Sam made an excellent point about the fact that there's sort of an opportunity to work in a module in the kidney because the kidney, each of our kidneys is comprised of 1 million nephrons. So there's a unit of the nephron that is comprised of the filtering part and then the tubular part that results in the final production of the urine that is put into the bladder. So there is an opportunity to perhaps take advantage of this. So understanding the module may help us build the whole organ as we go along. There was another point that was made about electron microscopy is actually quite surprisingly standard for every clinical case where we do a biopsy of a kidney in a patient. And so we actually have great understanding and a wealth of information about electron micrographs. So at that resolution of being able to see cell-to-cell interactions in that spatial manner. And so perhaps we could be mining that data and even going back to those EM samples and getting additional sort of measurements to recreate the spatial relationships. There was also some discussion, and I know some other groups had that discussion as well, about the opportunity to take advantage of bulk data in order to annotate single-cell data and sort of integrate these data sets for the benefit of being able to better understand, again, what constitutes normal and getting greater granularity as to the different cell types. And so that was, I think, the bulk of our discussion on the computational side.
We then moved on about validation, and I think we all agree that to some degree that depends on what it is we want to validate, what technique we use, but one needs to use something orthogonal and we need to look at an independent cohort in one way or the other. And that's validating the RNA signal, but then also moving on from that and see whether that translates into function, and then use surrogate functions such as proteomics, metabolomics, and then also use that unique opportunity with the kidney that obviously the kidney is a site of physiology, and try to interrogate that physiology, for example, in this organ-on-a-rig system where one can manipulate a kidney, insult it in one way or the other, and then get a transcriptional readout of what that insult does. There's also the unique opportunity to sample the contralateral kidney in organ donors as a direct validation. And then finally, we should really utilize the clinical data that one gets with every patient. So not only age and sex, but also what the baseline kidney function is doing in blood pressure and so forth. So all the relevant parameters of kidney function.
So at the end, we sort of asked ourselves, well, what do we want out of a Kidney Atlas 1.0, the first version? And so I think that there was some consensus about the fact that we really need to address the variability between samples. And there were some ideas about how to do that. Some of it came in the experimental part. Some of it is in the study design. The idea of showing preference toward perfecting our protocols that we can all share for frozen tissue so that we can actually more easily create cohorts rather than being, we're able to run cells, but those come as they come whenever there's a procedure and tissue becomes available. So if we're going to create cohorts and more systematically study human kidneys, that may be an advantage, and specifically because we are interested in obtaining data across the lifespan, so from fetal or early childhood pediatric cases all the way to adults and then older adults as well, so the effect of aging. Of course the effect of sex and race as well. And that's where we also thought that there may be some opportunities taking advantage of some existing structures that are there where there's samples being collected in other parts of the world, including Africa and Asia, so that we may have a more representative sample from which to draw. There was also the idea of gaining better spatial understanding by performing, for example, micro-CT or some other such modality that will give us a better picture of the entire kidney as we embark on understanding a small piece of it at the single-cell level. And then also the idea of integrating other existing imaging modalities such as histology or even electron micrographs, which, as I said, is sort of a unique feature of how we look at kidney tissue even in our sort of routine clinical care. And so these are just some of the first pieces that are coming together as to what we think would constitute the first attempt or the first version of this atlas. And I guess for next steps, we thought that we need to continue our dialogue, and so we hope to have a Slack channel for our community where we can actually bring in some additional voices. So one of the things that was on the earlier slides, we do think we need kidney pathologists to chime in because they have a much better understanding of the spatial organization of the kidney and the intricacies of the different parts of it. It's a very layered structure, very much like the brain with a cortex, a medulla, and many intricacies in those structures. And so perhaps input from the renal pathology community would be good. And I certainly know that I gain a lot from interacting with Astrid Waynes at the Brigham, who is the pathologist working with us on our projects. I think it will be valuable for the community. And I know that KPMP and other groups have also benefited greatly from having input from renal pathologists around the country, so I think that's important. So the Slack channel will allow us to engage with other members of the community, start to also look at sharing markers for different cell types, or starting to agree on these discussions between cell states versus cell types. And also, it's an opportunity to interact with the other communities that have ongoing work in this space. And I think it is wonderful if there could be some synergies in how we integrate metadata or clinical level data with our sample collections, a way to have a reference of where, which part of the kidney a tissue was obtained. These may be problems that are being solved in other communities as well. And so it will be important to be able to problem solve together and learn from each other. And so I think those were really our main points of discussion. And I just wanted to invite either members who were in our group or anyone else to really ask any questions at this time. Yes.
So I have two related questions. One of them, you mentioned cell types versus cell states twice. So my question to you is, what does cell type mean for you, and what do you want to use it for? And more generally, you gave an outline of what you want to collect for Atlas 1.0. But my question is, what would you want the Atlas 1.0 to answer for you? What would you hope to learn from it?
Mm-hmm. I mean, I think that anyone from our group could also chime in, but I think that the idea is to create the comprehensive catalog of all the different cell types in the kidney. The old, sorry? Defined types. I think that would be just all the cells that comprise a nephron, the functional unit of a nephron. In addition, it would be the immune cells, which we had quite a bit of discussion on. There may be resident immune cells that we could be interested in, even in a normal kidney, that may then inform disease states such as lupus, for example, which was discussed in our group. There's, of course, the vasculature. So there's many other cell types that are important. Endothelial cells are hugely important, and they're quite different in different parts of the kidney as well. So it will be important for us to understand that. Understand the cells that are important as comprising the nephron in the cortex versus the medulla of the kidney, and how they come together in the renal pelvis. So there's this spatial information that is important as well. But if you look at some of the old the old kidney physiologists microdissected nephrons and studied, the different parts of those. And so there's available information about different cell types from a long time ago, from many decades ago. And there have been, of course, many additional efforts to do this. And so we're looking at, upwards of, 26 more cell types in the kidney that we ought to be able to define. And in addition to that, we have all these other, pieces to it that we have to sort of assemble together. And so I think that we're getting there. We're starting to get there. My thinking about states, and this is something that the community needs to agree on, is states that may reflect different physiological perturbations, so to speak. Which is, the kidney responds every day to all kinds of different things in the process of filtering and, adjusting things like blood pressure, adjusting things like, hormonal regulation of various things. Secretion of erythropoietin is done through the kidney. Bone mineral metabolism is controlled by the kidney. There are many states that one could imagine one could capture, and that's where multiplexing data from proteomics and metabolomics, especially metabolomics, could potentially be helpful. I think that we're just scratching the surface of that, but that could be informative of states in addition. And of course, that's not even going into disease, where now you have major interactions with the immune system that we need to understand, and again, some ongoing efforts in lupus and other places, and I think that that's just going to build from there. But being able to understand the fundamental composition of the nephron and its interactions with other cell types around it that are still part of this integral organ, the kidney, like the vasculature and the immune system that's there, for starters, I think will be important through the lifespan and across gender and sex. So I think, we already have some ideas that, for example, men and women have a slightly different composition in the proximal tubules of the kidney. It seems that that's, from clinical information, that seems to be true from just basic histology. So wouldn't it be fascinating to be able to really characterize that at the level of the single cell for our atlas? I think that would be fantastic as a goal, as a first 1.0 goal, and with much more work to be done afterwards. Any other questions or thoughts? Anything I missed from any of the people who were there? Anything that?
If not,
Thank
thanks, Anna. Thanks, Tim. Also for opening the logo competition. So on the next HCA meeting, we have a logo breakout session for logo design. The next is Gary on the reporting on the liver.
Okay, great. So I'm going to tell you why the liver is interesting. I don't know if it's we could say it's the best, but the liver is not just a big bag of hepatocytes. That was one of the comments that we got in the last one of the last HCA meetings. Some interesting things. So interestingly, liver is formed of repeating units. This sort of hexagonal lobule here is repeated about 1 million times in the liver. It's about 1 millimeter or so in diameter. And then there are small capillaries in here that do the work of filtering the blood that are aligned with different cell types and have a fairly well-defined structure. The liver is a metabolic factory, over 500 functions known. It's an immune center. Most of the macrophages in the body are in the liver. It's got an amazing regenerative capacity. You could remove most of it and it will grow back. And one of the interesting things that I learned interacting with the liver community is that there are major liver diseases that are increasing in prevalence. And for instance, non-alcoholic fatty liver disease, or NAFLD, affects 20% of the world's population. It's like 1.7 billion people, and it's just getting worse with diets and issues with how people eat, etc. Viral hepatitis affects hundreds of millions of people, and so this is pretty. And there are no treatments to end-stage liver failure other than transplant. So. And the rate of these diseases as they're getting bigger is going to lead to more transplant needs, but you actually can't solve the problem that way. So we need to understand the liver to develop therapies. Okay, so there are a couple of maps have been published. The Itzkevitz lab published 2 groundbreaking maps in mouse over the past year, and Sonia McParlin, Ian McGilvery, and myself published a human liver map just last week with 5 human liver samples. And so these are out, so that's great. So we have our first draft map. The liver community is excitedly coming together. There are 9 liver projects that we knew about before that we're already talking to. All of these people are talking to each other. It was great that everybody, at least one person from every one of these projects, was able to attend this meeting today and is discussing about how to move the field forward. And we identified additional projects as well. So there's about 10 different projects that are active for liver mapping, representing a range of disease types and cell type interests. There's a fetal map. There's a nascent liver map project that hasn't collected samples yet, but as we identified samples, and the rest of them are focused on adult. Some of the interesting discussion points that were raised at our meeting this morning is that different people are working with different types of tissue collected in different ways. So we've worked with the transplant team to get a whole caudate lobe, and there's a protocol that is useful for that to get to that, was used for our map, but other people are working with biopsies and normal tissue, adjacent normal adjacent tissue from tumor resections, and everybody's developed their own protocols, so there's a big need among our community to compare these protocols, but we also realize that different people have different cell types that they're interested in, and these protocols are going to be different for different cell types. So we realize that there's not going to be one protocol that we're all going to use. There are going to be multiple protocols, and they'll be tuned for studying different cell types. So for instance, immune cell types are very robust, it seems, and hepatocytes die at the slightest, the slightest bit of processing. So there needs to be, most protocols will work maybe fine for immune cells in the liver. There are some specific challenges that people are facing with experimental processing of the tissue. As was mentioned for cardiomyocytes, liver hepatocytes sometimes have 2 or more nuclei. There's a huge mitochondrial level, like 2,000 mitochondria per hepatocyte, and these need to be, and there's also fibrotic liver samples that need to be dealt with separately. So one of the interesting ideas that came out of the discussion was that a lot of people have been talking about cell types and cell states from a biological function point of view, but we also need to think about this cell types from a practical experimental point of view. People were interested in knowing how they can identify whether they've got, how well their sample prep is working. So it would be nice to have a database, for instance, of experimental signatures that people see are associated with certain cell types, like certain transcript types and library sizes and other viability rates and other things like this that could go into a database of experimental factors that people identify. So when you do your analysis, you can know whether you are your rates of viability, for instance, are in line with what other people see on a per cell type basis. So so Dana Pear mentioned that there's that might be an idea for a portal to look at a range of signatures that are various different views of signatures of cell types. We also, it's absolutely important because of all of these problems with different cell types to look at single nuclei sequencing, and so one of the big things that we'd like to do as soon as possible is compare single cell and single nuclei maps. We realized that many of the samples that we get are inflamed, so there's a discussion of healthy, normal, and what we actually get from tissue samples. Neil Henderson was mentioning that there's ischemia effects, and so we need to we very likely need to look at model organisms for to help define what's normal and what's healthy, and be able to control the rate of inflammation or response to drug toxicity that might be actually widely prevalent in the human population. So I like what was mentioned with a couple of points earlier about how we need to study the disease situation to understand the healthy. In this case, it makes a lot of sense as well, because the healthy population is actually maybe frequently inflamed. So maybe half of the samples that we have are actually inflamed at a normal amount. So we have to study the extreme inflamed conditions to understand the inflamed factor, and also model organisms. Spatial is important. Liver for decades has understood that gradients are vital to the function of the liver. So as the blood flows through the capillary, there's a gradient of functions, and the cells actually act differently and have different gene expression profiles. And so we need to incorporate spatial technology. One of the questions is, we have 1 million repeating units. Can we just study one, or do we need to figure out if they're different in different places in the liver? The draft answer that we felt is that no, we can just study one for now, but it's not well known, so we do need to check. But there is preliminary results suggesting that the lobules are pretty similar across the liver, which is very nice, because that means that we can study this one section and extrapolate it across to the whole tissue. So it would be really great to have a 3D model of the liver where we know we've mapped all the vasculature, which I like the point about the vasculature coordinate system, and then attach these lobules to the vasculature and make a full 3D map. So that would be great. Dana mentioned that we could share technology with the Space TX project, so we'll be looking into that. We need to link with the immune map because there's a lot of immune cells in the liver. One of the interesting things that was raised is, we've talked a lot about the immune map. Immune cells are present all over in many different tissues, but endothelial cells, which are important in the liver, are also present in lots of tissues. So maybe we should have an endothelial map, even though that's not about liver. Endothelial cells are very important in the liver, and it's just an interesting idea that came out of our discussion group that could be food for thought for this community. Finally, just another practical little point was that a lot of people are developing site-seq antibody panels, and there are some standard ones that you can buy commercially. But we felt it would be nice if we could harmonize, share what panels people are developing and using, and even there was an idea that you could harmonize the antibody barcode. So if one person uses an antibody, if multiple people are using the same antibody, they all use the same barcodes. And so there was some discussion that could maybe happen through companies, but another thing to discuss in our community. So finally, we got agreement to share 10 new protocols on protocols.io. So there are 2 liver protocols that exist right now, but around 10 people said that they're going to post their protocols in the near future, so that's great. We also decided that we're going to work together on a combined analysis and map once multiple data are published. And also there's a lot of interest in forming a protocol comparison working group. So it would be a great idea to have a protocol comparison paper that comes out of that. And then we didn't discuss it in our discussion. I forgot to mention it, but other discussion around this meeting has suggested that maybe we should organize a single-cell liver conference, and there was some discussion in the organizing committee about the importance of these. So that would be, and everybody would be excited about that. So we're already thinking about how to do that. So Maz Hanifa, Neil Henderson, and Sonia McParland were the other leaders of our session, and Martin Williams and Charlotte Scott were the scribes. So if I missed anything, please mention that in the question session. Thanks.
So a lot of the groups throughout the past 2 days have been talking about making sure you get your right type of cells, looking at viability, looking at cell viability and cell fraction. What I haven't heard is making sure that your cells are unaltered and that the protocol doesn't alter your cells, particularly in the case of immune cells. You can keep them alive and happy looking or alive looking, but particular handling actually changes their gene expression. So I think viability and live dead should not be the only criteria by which we measure a protocol is good for a cell type.
Absolutely, yeah. So we need to catalog these ideas and, define what we need to measure.
About the lobules and the repetitiveness, because that was a question that also arose in the gut session and in the kidney session, right, on the repetitiveness of the units. So you mentioned that there's some preliminary notion around this. I think it would be very helpful across the board to get some answer around questions, and it might not port from one tissue.
So there's some discussion that work in mouse has shown when you take these lobules from different lobes of the liver, the gradients and their gene expression patterns are the same, basically, so from the Iskevitz group. So that's the data that I guess we have so far. So that would suggest that we could but there is some information that comes from liver disease that suggests it might not be exactly like that. So people know that the liver disease, certain parts of the liver are affected more preferentially in certain liver diseases. So that might suggest that it's not exactly uniform, but as a first draft, I think we could say reasonably well we would just study one and
But would you benefit if they are actually uniform or not? Would you benefit more from doing one, or if they're all the same, you could sample less from any of them, and you would still get sort of the same answer, but you might also then identify some variation that right now you're not, possibly is not there, but possibly is underappreciated.
So I think,
I'm just trying to figure it out.
So personally, I think it would be great to study the lobule very carefully, but
maybe spread some joy around?
So we could, but then also, as you just said, we would want to we want to check that things that we think are constant, look at the variation of those things. So you could do that in a few different ways. You could study carefully lobules from different parts of the liver, and you could also study a lobule, and we'll have information from multiple different samples no matter what. But specific features that you identify, like a gradient, gene expression across a gradient, does that feature hold in different places?
Yeah,
or the cell type composition, does that feature hold in
right,
different places?
because the gradients could hold in, right, within a lobule you could have the same gradients per Shalev's work. That actually does not rule out the possibility that they're still different from each other.
Yeah.
Right?
Yeah.
So I think that's the piece that we have to be careful
about. But we felt that, yes, it's important to figure that out, but it doesn't hold us back from going deep on one and trying as a first draft, say, this is what we'll extrapolate from one, but then let's check and we'll update. So I think that makes sense for us.
Yeah, I mean, you can imagine factors where some of these gradients, which involve particular genes and processes, are very repetitive for this factor, but then there are factors that determine differences across the organ, and you could imagine both of them coinciding.
Absolutely. Any other questions?
No more questions.
Okay, thank you.
Big applause to all the speakers.